The origin of the influx was at the early days of the Human Genome Project, when we thought that sequencing the human genome would be most efficient if we could separate all the human chromosomes and then sequence the genome one chromosome at a time. In the end, that turned out to be a fallacy, but it did motivate us to build a sorter that was more precise, because the human chromosomes are very dim and very small, so it's not easy to measure them accurately in a flow cytometer. And for that reason, we made a very flexible modular illumination system, a modular detection system.
There is just the modular nature of the BD Influx in that it accommodates the scientist who has a very simple experiment that involves one color and one laser, up to the person who's doing 7 lasers at a time and doing 24 colors, 32 colors, whatever it is. The advantages to having the 7 laser sorter is that every month or two we get a new flavor of laser, you get new fluorophores, you get new assays, and the assays are becoming more and more and more complex.
With the many laser colors that are available, you're not limited to a given set of fluorescent dyes anymore. You have the freedom now to select whatever excitation color you want, depending on the dye or dye combination that you are using in your experiment. One of the characteristics of the influx system is that it has very sensitive low noise electronics. In order to measure the height of a pulse, we also need to know exactly where the floor is. You can't measure the height of an object if you don't have a stable and immovable floor. So in order to keep the noise, keep the ground level of the system as accurate, as stable as possible, we use analog electronics in our first stage of the signal amplification. So at the heart of the influx electronics is a unique digital architecture that knows when the particles go through the separate laser beams, keeps track of the timing, and makes sure that the digital values that are stored in the system all belong to the same particle. And it can do that while as multiple particles are going through the laser beams. So we may have a particle that is being measured at the last interrogation station while there are already 2 or 3 other particles being processed at earlier stations.
As cell sorting has evolved, it's a fairly simple idea just to, instead of sort one way, 2-way, 4-way, to do multi-way sorting, so that you have the ability to capture any of the populations that you see that you find interesting, so that you can separate them out either for later study or to grow them up or to do some more science on them.
The Influx platform is designed for research applications that haven't been invented yet.